Good day. Welcome everyone to the Rigaku virtual event and the roundtable discussion on polymorphism of pharmaceutical actives. I'm Simon Bates, and I'll be the moderator for this session. Before starting, I would like to personally thank all the roundtable members for their participation and you all in the audience for attending. Thank you. When we first started to think about a virtual conference highlighting the role of analytical within the pharmaceutical industry, we reached out to the community, and the most requested event was a roundtable discussion on polymorphism. So that brings us all here today. The roundtable members are all from very diverse backgrounds, and during the discussion, we'll hear about past, present, and future perspectives of polymorphism and the pharmaceutical industry from some quite different viewpoints. So to start the ball rolling, I'd like to invite the roundtable members to introduce themselves and briefly outline their historical perspective with respect to polymorphism, essentially what it is and why is it important. So Steve, if you'd like to start off.
Hello, everybody. My name is Steve Byrn. I'm a professor at Purdue. And my introduction to polymorphism started actually 52 years ago when I was a grad student at Illinois. And my major professor had found in Biostime a report of a yellow to white conversion of what the author actually said was a conversion of two different compounds. But so we started investigating this and found out that there was a yellow and white polymorph. The white polymorph was the most stable. The yellow one would convert to the white one on heating and up front. There was actually front movement through the crystal, and the rate of front movement depended on the temperature. It was pretty unique behavior. And so we set out back in the day to do single crystal structure, and we used Dubai shear cameras also for powder. And so we were able ultimately to get a crystal structure of both forms and publish most of that in JCS. And that was not a drug, that was a terephthalate ester. But that keyed my interest. And when I got to Purdue Pharmacy, then we started looking at drug behavior. And a lot of the drug work, I actually piggybacked on NIH grants for cancer research. And so we continued doing that research, and we worked with most of the people here. Ken was at Purdue for several years and was at SSCI. And it's just been a great run to work on polymorphs. I had a lot of fun.
Yeah, thank you, Steve. Suri, would you like to follow on?
Sure. My name is Raj Suri, and I've been teaching at the University of Minnesota for over 35 years. Now, my broad interest is in the field of pharmaceutical material science, and my group has been interested in developing new analytical techniques for solid phase identification and quantification. And we have successfully quantified different physical forms of the active ingredient in complex formulation matrices. And when I say the term physical form, it encompasses polymorphs, solvates, and crystalline and amorphous states of a compound. We are also interested in monitoring processing-induced phase transformations, specifically as a result of, say, granulation, milling, compression, freezing, and freeze drying. Now, our most recent work, we have spatially mapped the phase composition in dosage forms, specifically tablets. And this has enabled us to monitor differences in composition in different regions of the tablet, for example, between the surface and core. And powder X-ray diffractometry has been the primary analytical technique. We have also used the synchrotron source at Argonne National Laboratory when enhanced sensitivity was needed. And differential scanning calorimetry, dielectric, and other spectroscopic techniques have also been extensively used. Now, coming to the point of why this is important, from a pharmaceutical and patient perspective, our concern is product performance. We need to establish the role of the polymorphic form on the final product performance. Moreover, control of the polymorphic form may be important from a process control as well as patent and intellectual property perspectives. And I want to acknowledge all the other people in the room here whose work has inspired us in many ways. Thank you.
Well, thank you, Suri. Anne?
My name is Ann Newman. I currently work at 7th Street Development Group. I started my career at Squibb, which became Bristol Myers Squibb. I worked there for 10 years. And then as Steve said, I worked at SSCI for 10 years. I was introduced to polymorphism in graduate school. My Ph.D. is in inorganic chemistry, and I did a lot of work with molecular sieves and catalysts. And I expected to work in the petroleum industry, to tell you the truth. And so when I started looking for jobs, I had a whole long laundry list of analytical techniques that were on my resume. And when I got to Squibb, they were just like, we want you to use those techniques on pharmaceutical compounds. And I was like, it can't be that easy. And it wasn't, but it was a great area for me to start in. And then I worked with Ken at BMS as well. And we ended up joining our groups a lot in order to look at a lot of different types of systems and solve a lot of problems. And I think the importance of polymorphism, as I've seen over the years, is people want to check off a box, but when they don't really apply it to all stages of development, it affects API manufacturing, crystallization, drying, storage, handling. As Suri said, process-induced transformations, again, storage and handling. Bioavailability, is it going to change in the GI tract? So I think it's one of these things where we think it's like this, but it really covers the entire range of development. And I think that's important for people to understand.
Thank you, Anne. Ken? You're on mute, Ken.
Yep. Yeah, I just did. I raised my hand, but you didn't see it. That was my name's Ken Morris. I am a retired faculty from Long Island University and University of Hawaii before that, and 10 years at Purdue before that, and BMS with Anne before that, and a lot of jobs you don't want to hear about. My introduction to polymorphism actually came after my bachelor's degree. I was working at the Environmental Protection Agency, and we were trying to equilibrate these really hydrophobic polynuclear aromatic hydrocarbons with sediment water systems. And sometimes you would do that and it would equilibrate over the period of a week. Sometimes it would take 6 weeks or never equilibrate. And my boss there was a solid state physicist, Sam Karakoff, and said, well, it's possible that you've got a different crystal form. So we broke the carboys and x-rayed them, and in fact we did. So that was my introduction. So that had little to do with pharmaceutics, of course, but Sam Yalkowski was from the University of Arizona, was the consultant who was working on modeling partitioning into stream sediments. So the Ocnall water partition coefficient work fell into that very nicely. And so I went to graduate school in Arizona, where I am now again, with Sam Yalkowski, and worked on polymorphism of impacting on solubility predictions. So that sort of led me to stay in pharmaceutics, and it paid better. So I didn't go back to EPA and went to Bristol Myers Squibb, where we had the material science group that worked between Ann's group and Abu Sarajuddin's group. And then after that, when I asked Steve for a recommendation for a job, he said, maybe you should come to Purdue. You know, we're taking in refugees. So I went to Purdue and worked with Steve and Ann again for many years, and was introduced to, of course, even before that, to people like, excuse me, like David Grant and Raj Suri and Mansoor Khan, although we never put the Raj in front of Suri, it's always Suri. And so that's sort of where I am. I've been focusing on mechanical properties more than solubility and physical, and chemical degradation, although more of that's coming in with the last publications that we've had from LIU. Thank you.
Thank you, Ken. Eyal?
Hi, everyone. My name is Eyal Barash. I'm a patent attorney, so a little bit different story than the distinguished people before me. But I did start out as a proto-scientist. As an undergraduate, like Anne, when she was in graduate school, I studied organometallic chemistry at Indiana University. I worked in a lab for several years there, where I became familiar with solid state NMR, I actually worked in a solid state NMR lab after undergrad and went to grad school in solid state NMR, but also IR, a little bit of Raman. So I knew a fair bit about analytical techniques. And I did actually, I never told Simon this, I did actually take an X-ray powder diffraction class when I was an undergrad, but I never could figure out what exactly K-space was. So I pass fail. Anyway, so I decided to go to law school, and I wound up in DC doing pharmaceutical patent law. And lo and behold, I heard about a case in our firm involving polymorphs. And I'm like, that's interesting. What's that about? And I was looking at the patent, and there were things about DSC and NMR and X-ray. And I told one of the partners, well, I actually know a little bit about this. And they said, you do? And all of a sudden I spent the next, several years of my life in that case. And that's how I got to know Simon and Steve and Ann and Suri and Ken and everyone on this when I was at, because then I joined SSCI. I was through SSCI and others and David Grant. I became the counsel for SSCI doing patent work. And eventually I started my own shop, which is where I am now. But I still devote a lot of my time to what I call solid state chemistries, whether it be crystalline forms or amorphous forms or others. But from a pharmaceutical perspective, trying to protect these forms because they're extremely valuable, especially from drug delivery, and also from the angle of drug repurposing and drug repositioning. Many companies are winding up with patents on their chemical compounds that are old, and they're trying to figure out a way, how do we reinvigorate the IP portfolio? And as they go through development, they realize, you know what, maybe I will make a mesylate salt of this. Then how do you protect that when you have a prior form that maybe is a free base? So crystalline forms play an enormous role in that. And there's a trend among pharma to take advantage of the new and innovative properties of crystalline forms to get new IP, breathe new IP life into drug products. So that's something that really interests me with respect to crystalline forms.
Thank you, Eyal. Mansoor?
Hi, good afternoon, everyone. My name is Mansoor Khan, and I am a professor and vice dean in the College of Pharmacy in Texas A&M University in College Station. And prior to my job in academia now, I worked in Food and Drug Administration as a director of product quality research and advisor to Center Director, who is now an acting commissioner of FDA, Dr. Janet Woodcock. And prior to that, again, I was a faculty in Texas Tech University. Now, my interest in polymorphism came when I started doing my master's degree. That's when I was looking at some of the solid dispersions and co-precipitates and how the product characteristics were changing with time, and how when they were exposed to some temperature and humidity particularly, how it was reverting back to their original crystallinity. And so I went to geology department, took some X-ray diffraction course, started looking at X-ray diffractometer, and that has just continued to grow and grow. And this was a part of my PhD work. And after that, I think down the road in some of those challenges, I can talk more about it, but I have seen a lot of challenges and a lot of problems that had to be corrected almost instantly, and which is not very easy when different firms are looking at it. And so we'll talk more about it later. So in Food and Drug Administration, I have seen a lot of cases involving solid dispersions or polymorphic changes, and how do you characterize them, how do you evaluate them, and how do you ensure that the product doesn't change. We'll talk about it. Thank you all.
Thank you, Mansoor, and thank you for those interesting introductions. With that, I'd like to lead off with the first topic, which is with respect to polymorphism, what do you think are currently the biggest challenges, and how should we address them? What should we start doing, or what should we stop doing right now? And Mansoor, if you'd like to start off with that.
Yeah, one of the biggest challenge we have is the product consistency. So you make a product, whether it's solid dispersion or any other technology, you made a product, and so you developed it through various phases of R&D, and then you made a product, but that product is being made consistently. And that product is being made at one site to another site to the third site, and then every time we have to get back to the same original quality that we had. So a lot of times the products fail, and when the product fail, when they are on the market, they get recalled. And if you see the recalls, there are more than 1,000 recalls of FDA products every single year. Starting, I looked at it from 2007 onwards till now, almost 1,000 or more products, right? So now the question is, why did it get recalled? Now, there are firms who have done a great job. Their products do not get recalled. There are firms who do not understand their polymorphs very well, who do not understand and who do not control their products very well, and who do not even identify those products very well, and likely that those products have failed. So they had some release testing, which means every time a batch is made, and they have to do certain testing, there is called a release testing. So they do a release testing, everything passes. But after some time, the product fails. So I'll give you an example. A product where a dissolution has gone down. Initially, it was meeting all the dissolution requirement when the dissolution has gone down. When the dissolution goes down, people give different explanation. Oh, my operator was not trained, or it wasn't processed very well, or it could be that, hey, there was this polymorph that's reverting back. But how do you quantitate? And if your dissolution method is not discriminatory, then you won't be able to identify that. So a good firm understands these things. They have a state of control on their products, but a firm that doesn't understand this polymorphism doesn't quantitate, and it's very likely their product changes. So I've seen products that have changed. I know of one specific example where a product was made, very good product, very widely used product. They just changed the site of the firm from one region to another one. For example, from India to Puerto Rico, or from US to Puerto Rico. They just changed from one place to another place, started seeing a lot of failures there in that case. It turns out that, the humidity and the environment might have played a very significant role in a case like that, right? So.
Yeah, thank you. Eyal?
Yeah, so, what I find in terms of issues with polymorphs and things that scientists can do is, I would say maybe glibly work with the attorneys. What I often find when I look at patents that are drafted in this area, typical chemist attorneys or patent attorneys who have a chemistry background don't know this space very well, and they rely very heavily on the scientists to help them. And one thing that I sometimes see is a scientist may say, well, here's a great X-ray powder diffraction pattern, and here's a table of the 75 peaks between 3 degrees and 50 degrees, and all of that winds up in the patent. It really is a collaborative process, because for every claim, for every piece of data you put in a claim, you have to prove that it's there in order to infringe the claim. So a great PhD thesis becomes a very difficult proposition for proving infringement. So something collaborative is very good. And one thing I think would also be is useful is that when I'm working with scientists to try to claim a polymorph, claim a crystalline form, one of the things I always ask is, how do we know that this is a pure phase? And so, for example, I'm a huge fan of indexing, and if I can get a single crystal structure, that's nice too, because it becomes challenging when you file IP on something that you think is X, and actually it's X plus Y. So whether it's X-ray or some other technique, what data do you have? How do you know, when you tell me that this is a new form, how do you know that it's a new form? And how would you be able to defend that? Because ultimately we want to be as clear as possible when we file on these patent applications, because unlike I would say a new chemical entity, we file on so many new chemical entities, but very few of them, quite frankly, wind up being valuable. If you're at the stage where you're actually now making the investment to file on crystalline forms, someone's already made the decision that this is of pretty big value. And because of the nature of the fact that you need so much data to describe these forms, you want to be very clever about how you describe the data accurately, but it's a collaboration with the scientist and the patent attorney. And the patent attorneys often it's a steep learning curve to get into this space.
Thank you, Eyal. Ken?
One is that there's one aspect of polymorphism that gets overlooked almost universally, particularly as you get into later early development and early mid development. And that is, what are potential patient-based failure modes? Because there are patients at the end of the line somewhere, so that if there is a compound whose form may impact whether it's solubility or manufacturability or propensity to disorder, that can have a therapeutic effect on the therapeutics, it's important to identify that early, particularly before you go into tox, of course. And there are not stated mechanisms for doing that, but we all sort of know the basic pathway, and some people have spent time sort of elucidating that. I think in terms of the next level, though, is the pharmaceutical equivalence issue, because pharmaceutical equivalence, as Mansoor and everybody can tell you, is a moving target when it comes to Q3 equivalence, whether or not something is actually microstructurally equivalent and therefore expected to perform the same and maintain its quality, as Mansoor had talked about. And also, I think adding to the challenge there is you really have trouble doing low-level detection in dosage forms. And sometimes, and Suri and I and Mansoor and Steve and others have worked on levothyroxine, for example. And it can be just almost impossible to get any data on that small amount of material that will tell you what the actual structure is, much less quantify it. And that's the next step, is how do you quantify? Because we know from the teachings of Simon and others that using standards like we use standards for an HPLC for solid state just doesn't work. You may be in the right ballpark, but you're not going to get to the level that is necessary for compliance issues or safety. And so given that, I would say that the relevant standards is a huge hurdle that needs to be resolved. And that goes along with getting structures of metastable forms, because a lot of the issues that come up are issues that are associated with metastable forms. So all of this sort of folds back into the large heading of pharmaceutical equivalence, with the goal of being able to anticipate patient-based failure modes, recalls, and side effects. So, thank you.
And thank you. Anne?
So I agree with everything that everybody has said. In fact, as a consultant, I've had problems with every single one of the things that you've talked about. And a lot of it is, clients want, as I said before, check off a box, and they don't really understand the impact of polymorphic forms. And, that's kind of where we all come in order to educate them on this. I think a couple of areas that I see is we rely on the same techniques, which may not be applicable for every system. I think branching out into new areas and new spectroscopies, new ways of looking at things, even looking at more computational type of methods, as Simon has been doing and Suri has been doing, we really need to continue these, because this is where we need to go in order to do our best work. The other thing that I spend a lot of time on with clients is the whole idea of, you can measure something in an instrument, but once you take it out of an instrument and you've exposed it to ambient relative humidity, all bets are off if you've got the same thing. And I think we need to understand that in situ methods are extremely important in understanding what goes on in these metastable forms, but we also have to have an understanding of what happens once we expose it back to ambient RH. And I see that as a disconnect in so many different projects. Even headspace in vials, I find a lot of people, they have no idea what the headspace in their vial is. Different RHs from 30 to 70% can occur in these vials. So I think there's a lot of understanding of conditions and how our forms may change under different conditions that we need to continue to look at.
Thank you, Anne. Suri?
So the first point I want to make is that looking at the drug substance may not be adequate, because the polymorphic form in the drug substance may not be the polymorphic form in the drug product, because processing and the presence of excipients can have a bearing on the final form. And identifying, let alone quantifying the polymorphic form in the final product can be a challenge if the drug product constitutes a low weight fraction of the dosage form, as Ken mentioned. And just to give you an example, let's say 10% of the formulation is the drug and 10% of the drug undergoes a polymorphic conversion. Now, the new form constitutes 1% of the dosage form. So we are very quickly getting into issues of analytical challenge, particularly when we are looking at this in the context of a complex formulation matrix. And also, though we are talking about polymorphic form, we need to look at the solid form comprehensively, because imagine a case where a drug also exists as a hydrate, and then during processing, it becomes anhydrous form, and the anhydrous form can now be a new polymorph. Now, similarly, if there is any amorphization of the drug during processing, it may recrystallize during storage and again to a different polymorphic form. To me, the amorphous form poses particular challenges because it means increased chemical as well as physical reactivity. Thus, if an amorphous phase is formed unintentionally, then we have a metastable phase, which can be much, much more reactive. You also asked a question, what should we stop doing, Simon? And in my mind, one of the questions that we need to ask is, does the polymorphic form control the product performance? And that product performance, I'm speaking very broadly. And if the drug constitutes a very low weight fraction of the dosage form, and if the polymorphic form does not influence product performance, then how much time and effort is it worth to control the form? In other words, what purpose is served by controlling the polymorphic form? That's a question that we as a community need to ask. Thanks.
Yeah, thank you, Surya. That's actually an important question that hopefully we can get to in the roundtable. Steve?
Well, like Ann said, I agree with everything everybody said. We, Pam Smith and I, actually started a new company called Improve Pharma, and we've had a number of projects representing some of these, and I think it's good to summarize them. One set of projects involves what Raj was talking about, conversion and interconversion of forms in dosage forms, and how you can measure that and study that. A sister of that is salt disproportionation. It's a number of us have published on that, but salt disproportionation, I think, is more common than we realize. Especially, it seems like if you have a very insoluble free base, as people talked about, there can be a tendency to disproportionate and form the free base, which will precipitate that, basically form a very insoluble solid product. And then we've been doing a lot of work on salts in the area that Eyal's talked about, salts. What are the opportunities for getting new salts? And how, what will happen with those new salts? Can they be brought on the market as alternative products? And then, of course, when you make salts, you have to determine the polymorphs. And that can be a huge project. And then the fourth area that we've been dealing with is small amounts of compounds. Very obviously, especially in early discovery, they're not large amounts of compounds. So sensitive X-ray equipment that allows you to look at small amounts. We've been doing synchrotron in some cases, but small amounts of material and technologies that will let us get decent patterns from small amounts of material, I think is very important. That's all for me.
Yeah, thank you, Steve. Yeah, because there's so many diverse opinions here, I'd like to open up the table for people to comment or answer back on other people's discussion. So feel free.
Thing I'd like to say, maybe following on Steve's point and some others, is that one of the things that we're looking for as attorneys to help our clients protect these forms are, what are the properties of a crystalline form, as opposed to the form, if it's a follow-on product, as opposed to compared to the previous one. So for example, what are things like the dissolution rate or the solubility? Or if you're putting together a co-crystal, does the co-former have any kind of medical benefit or something like that? Because in the United States, it's generally easier to get a patent on a crystalline form by relying primarily on the difference in structure, whereas in other jurisdictions, you really need to show some kind of benefit. And really the best people who can help us determine either what the benefit is or to devise experiments to help interrogate what the benefit may be are people like yourselves and people who've been trained by you. So I would suggest that, that kind of thinking in terms of the practical benefit is of great import. And I know when you think of like, how does this affect the patient down the line, that's where these two systems sort of meet, the IP and the clinical benefit.
I think, sorry, I was going to say, I think the point which Anne and Ken made about the impact, that should be the key for the products that we make. Eventually, it's the patient that's actually taking, you see, consistent, he or she getting a consistent product throughout their lifetime or not, because patients are in chronic condition using for a long period of time. If a medication doesn't work, what happens is they tend to double the dose, or they tend to increase or decrease the dose if it doesn't work. But it turns out that after some time, another product comes and that really works. And now what happened, if you increase the dose, that's too much of the dose for that patient. So that's why when they are recalls for the product, right, they are class 1 and class 2 and class 3, depending on the severity, on the impact on the patient, right? Those recalls are there. Some things that are coming to my mind, as you all know, is that, I work in the formulation science area. One of the key elements of formulation science is the pre-formulation, where you're actually characterizing and stabilizing the API, and then seeing the impact of the excipient, the process, the environment, the stability when you are making a product, right? So all these things need to be considered and understood well so that the product doesn't change. Now, in terms of polymorphism, one of the challenges we have, so you're looking at X-ray diffraction, and so you have some peaks coming out, some 2 theta values that are coming out. First immediate thing is, if it is too low a level, then you are not able to accurately characterize it. Now, suppose you are able to do it. Sometimes people have done it with 5% or even down to 1 or 2%. People have been able to do it for certain drugs. But like levothyroxine or examples like that, they are so low, it becomes very, very difficult. On top of it, you have excipients, and that are also contributing to 2 theta values now. So now you need to be able to separate them out, which is coming from excipient and which of these peaks, and which is coming from product, and which of them are consistent, which of them are changing. And another challenge that we have is the destructive method. Sometimes you have a method that, you have only 3 samples, 2 samples left. This is a criminal case, involving some big loss somewhere. So you only have 5 tablets left, or 10 tablets left, or 3 tablets left. And if you use up those tablets for this analysis, there's nothing left as evidence there, right? So if there could be methods where it could be non-destructive, that could be very helpful. And the impact of it on the patient, as well as the impact on the formulation in vitro, if you're able to detect. So your method has to be really able to discriminate between polymorphic changes, one polymorph versus another, or the same polymorph if it is changing with time or with environment. Distillation method should capture that. If it doesn't, then the product has changed, the patient is hurt, but we won't even know it's because of the polymorphism, right? So, and lastly, one more point I wanted to say, sometimes we rely on a single techniques. It could be a challenge because of all the interferences levels, and also we should be able to corroborate with another technique. For example, are there methods with DSC, or are there methods with near IR, or something else that we can corroborate these results, or for example, the dissolution that you have. So I thought these are some of the things I just wanted to highlight.
If I have to emphasize that, I think the idea of orthogonal techniques. I know Steve had pioneered some of the use of Raman in addition to X-ray to be orthogonal to get other eyeballs on the same sample, which really works well. And the one thing I wanted to highlight, though, was in the discussion from you, Mansoor, as well as Surya, is that this idea of process-induced transformations, which we've all known about for quite a long time, really is almost the fly in the ointment with respect to, in the negative sense, a fly in the ointment with respect to analysis, because as we talked about before, it's even if you assume that you have all the analytical power that you need and you can detect things at the level you need to detect and quantify them, it has to be done throughout the development process, and it has to be done throughout the scale up, so even if you can do it at the API with the Hilo lab in full scale, then you still have to do it at every step during manufacturing. And when they change, so Abu Sarajuddin used to say that, pre-formulation was the center of the universe. And I think it's not too far a stretch to say that certainly at least you should have a cycle back through that. And I know Ann's group specialized in that at BMS when we were there.
Yeah, but following up on what Mansoor, you said, and Ken's remark, see, when we compare, polymorphic forms, say, from two different manufacturers, my problem is that we are always comparing, we are looking at the composite effect of formulation and form, right? So how do we tease out the physical form from the formulation? If you recall, the most pioneering work was done by Armando Aguiar in Pfizer, 50 years ago on chloramphenicol, where the stable form just did not give blood levels. It didn't get absorbed at all, so it was black and white. But my hunch, again, Mansoor, you know this firsthand, my hunch now is that the differences are much more subtle. And how do we as a community distinguish the formulation effect from the form effect?
That really throws open a big question there. The way we we try to look at that impact through bioequivalent study, especially for a follow-on for a generic product. All we are doing really in the clinical paradigm is doing a bioequivalent study, right? So in vitro, you're doing all the work to see dissolution changes or not changes, but I will select a media with some surfactant in it and that kill all the discrimination ability. And then so it doesn't show any difference. And then I'm doing a bioequivalence testing, and then bio just matches, and that window is so broad, right? The confidence interval window is so broad, it just passes. But the problem is, in the real life, patients are taking this medication for a long period of time. Things change, and that's why there is no way to actually capture it, because our regulatory paradigm doesn't allow us for these bioequivalence tests with time, like stability tests you're doing with time, dissolution you're doing with time. The bio doesn't do it with time. And that's the reason you get recalls. And then there are lawsuits and there are other problems because of that. So,
but bioequivalence is no better than the pharmaceutical equivalence. If you don't have pharmaceutical equivalence, then bioequivalence is whistling in the dark.
Exactly. But the way pharmaceutical equivalence is defined, everything is pharmaceutical equivalent. So if you have an oral dosage form versus oral dosage form, 5 milligram versus 5 milligram, right?
Yeah, but it's the Q2. Q2 is a minimum. Q3, which doesn't really exist outside of Rob Weinberger's formalism.
Right. But pharmaceutical equivalence, Q2 for excipient is not there. It's not required.
Exactly.
Only the drug API is required to be the same strength, right? So that is a fundamental problem there, right?
No, but I guess I'm saying, Mansoor, is that pharmaceutical equivalent is insufficient as defined now. And that's
absolutely
that's part of what I was going to say in the future stuff. But that's, if we can't come up with better criteria for pharmaceutical equivalence, then I don't know what the future is.
So you have polymorph A versus B, the example which Suri presented, chloramphenicol, A, B, C, right? They're all the same for pharmaceutical equivalent. It doesn't discriminate between A, B, and C, where the bioavailability is different. That the responsive patient is different, right? But pharmaceutical equivalent doesn't capture that. And you're right, Ken, that's an area that needs to have discussions in the community, and that needs to be improved, really.
So, Mansoor, coming back to this, in case we identify top 5 or top 10 challenging compounds, what stops FDA or the broader community from mandating some bio studies on these compounds at the end of their shelf life? I mean, just to look for, is there a problem?
For that, you have to show documentary evidence that it is harming the patients. And so that's when they will do it for that particular product. But to have a broad changes for all the dosage form to do bio studies, already there is a huge a problem of the expense that is needed for the development. It adds a lot of expense, especially and just the beginning and the ending. And what about other times and beginning of ending, and what conditions? Expiration date. So do you want to store it for 25, 60 or 40, 75 and then wait until do that study in that one condition? Then the next question comes, during storage and transportation conditions change. And then another question that comes is the patient is taking this product in a pharmacy vial, whereas we did all those studies in the original container. So these questions keep coming.
And the criteria for bioequivalence is pretty broad, and the people change, and so.
Yeah, thank you. In the interest of time, oh, Steve, did you want to say something? Or?
Well, I wanted to pick up with a couple minutes on some of the things Eyal was saying and some of the things we're working on. The purity, finding the forms and confirming purity continues to be a big problem. I agree with Eyal, indexing. I'm a big fan of indexing because it's going to allow us to verify that when we're doing screening, that diffraction pattern is a pure phase. We also have started doing some micro screening where we actually fish out a crystal and go and get a single crystal. And then we can compare that to the calculated pattern to the observed pattern. And we can start to figure out. And what we've seen in salt screening is a number of cases where the free base shows up and complicates the screening. You might have a salt, but you also have a free base. And surprisingly, we saw that it was worse with the hydrochloride salt. And I think it's due to the volatility of the HCl gas. But the hydrochloride salt was actually prone to form mixtures of the free base and the hydrochloride salt. It's very difficult to work with. And some of the other salts, we published some of this, like the benzoates, were quite much easier to make salts. And so some kind of mixture of well play, fishing out crystals when you see them, and powder diffraction, and working with small amounts of materials and microscopy, back to Walter McCrone's methods. I think these could be paths forward in early screening.
Yep, thank you all for those comments. Anne, did you want to say something, or should we move on to the next question?
Well, I just wanted to add one more thing that people were talking about process-induced transformations and those types of scenarios. And what I have seen so many times is a client will do one screen early in development, they pick their form and they never go back and look at it again. Mm-hmm. We have so much information in those screens about possible transformations, either from crystallization, drying, especially product development, product processing, and we never go back and look at it. So I really think we need to start looking at our screening data more closely every single time we're looking at a process. And I think that's missing in a lot of development programs.
Yep, thank you all. So to move on to
real quick, we had a case where apparently from the patents, there was a single salt form, then a Czech group patented 4 salts, and then we investigating, we found 7 more salts. So like Anna is saying, there's people may be missing quite a few salts, especially in certain areas. We're publishing some of it because it was a project with Gates, but it's quite interesting to see some of the capability, some of the things that can happen now.
Okay, thank you all. Let's move on to the last question. So to wrap up the roundtable discussion today, I'd like to ask the members to express their opinion on future perspectives this time. So projecting a few years down the road, do you see the role of polymorphism as an evolving one or are there some revolutionary breakthroughs on the horizon? I'd like to start off with Steve for this, and we'll go around the table.
Yeah, I think it might be evolutionary, not revolutionary. I think certainly the working with small amounts of material. I think single crystal and indexing have a lot of potential, and I think synchrotron. But I think it could be a combination of X-ray methods are going to advance over the next few years and give us a lot more capability to do what Ann said, to figure out what's happening in these screens and how many forms there are and the most stable forms.
Yep. Thank you, Steve. Suri?
So I'm going to make two assumptions: that the oral route will continue to be the most popular route, and that more than 50% of the drugs under development belong to BCS class 2. So since the solubility is the limiting factor, controlling the physical form provides an avenue to modulate the solubility. So from this context, the physical form, and specifically the polymorphic form, will continue to be important. I think that the analytical techniques will be getting better and better, and therefore the limit of detection and limit of quantification will get lower. The last point is, as the drug candidates increasingly have undesirable physicochemical properties, we will be relying more and more on excipients to make the formulation effective. And so the functionality of the excipients take on an important role. And so in addition to drug polymorphism, issues of excipient physical form are going to become more important from the context of their functionality.
Yeah, thank you. Ann?
Yeah, I guess I see it as evolutionary, because I think polymorphs and forms, we're discovering so many new things in pharmaceutics. You know, we went from amorphous to amorphous dispersions to now coamorphous, and we keep adding different things. We go from binary to ternary systems, we're in quaternary systems. So I think I see it evolving as we get more creative, and, you know, maybe necessity is the mother of invention. But so I think we're never going to end with the number of different kinds of forms that we're eventually going to find. From a revolutionary point of view, I would love to see more computational ways to at least get a handle on things, you know, even if, you know, so I can try and either reproduce it in the lab or compare it to what I've already got. To me, I would love to see that, because I think in so many ways it would make our lives easier to understand the crystallization landscape that we may be able to either attain, or that we've already there and we don't even know it. So I kind of see it as both.
Thank you. And Ken?
Yeah, thanks, guys. So yeah, following up on what Ann said, I think there are a couple of things that could be evolutionary, revolutionary. I'm not really sure. I'm a tactician, but the use of big data for structure solutions is really an underdeveloped area. I mean, they've done a fair amount at Cambridge, but that's not their primary function. But that'll get you not only new forms, estimates for new forms, but also estimates for variations in forms, because part of the problem is that you never have an ideal crystal, even singly, much less in a population. So I think that's a big gap that's going to be filled in building it into the computational realm. That's where the biggest advances for Dassault systems and others have come from, is taking the computational methods and marrying them to the big data, and at the risk of sounding trendy, AI approaches to actually solving the structures. The one thing that would help that a lot is to really facilitate the sort of lab instrument levels that could approach synchrotron resolution. I mean, I think if we could do, or intensity, if we could do that, I mean, there's the benchtop synchrotrons and things, but I think that's a big area, and that's under development in several places. I think, Simon, you can speak to that more than I, of course. The final thing I wanted to say is that the studies that we're talking about are only going to become more important, to Suri's point. They're only going to become more important because not only are the generics going down the road of developing dosage forms without having the prior knowledge, hence the term we've been using, new prior knowledge to generate the kind of information they need. But now with repurposing, this is even more critical. Without this, we're in big trouble. We'll have more and more recalls, more levothyroxines and warfarins, et cetera. And the last part I would link that to computational as well, but is the sort of the computational PK, and that is for reformulation. Most recently, reformulation for geriatrics. I'm the poster child for geriatrics has become really problematic. So people are having all sorts of problems with these formulations. Mansoor's probably heard about it from the agency, but and they can't. The things that worked when it was a simple tablet just don't work when you put it into a different dosage form. So I sort of see those as the areas that I would stake some money on. So.
Yeah, thanks, Ken. Eyal?
Yeah, I think that computational work, AI, machine learning, again, whatever code word you want to use that's trendy, I see those changes now, and they're going to have some big IP implications. Recently, there's a case that's been working its way through the court system called Davos. It has nothing to do with crystalline forms, but it has to do with the concept of what happens when an AI comes up with an invention and there's no human invention. In fact, there's perhaps some already analogies with respect to robotic screening, where someone has programmed the robot, but it's the robot that's doing the screening if it's fully automated. So there's some challenges I think that are coming down the pike with respect to not only who's an inventor on new forms, depending on how it's done, but also what happens when you reach the stage where we can say, well, hey, we can actually tell you in advance what the stable form looks like. How does that affect the patentability of these forms? And so I think there's some challenges coming up, and I think clever people are going to come up with some solutions, but it's going to be an interesting road down the line.
Yeah, thanks, Eyal. I agree. Interesting times. Mm-hmm. So, Mansoor?
One other thing that I'm thinking is the disease management. There are certain diseases. For example, you have brain tumors, or you have some very critical situation. What role does this polymorphism has? If we, for example, if you go to NIH, look at some study section, people talk nano and nano and nano, there's a lot of role for it, right? So because it's really going into areas, some tumors that in other ways it's not able to go. So do we have some polymers that can go, or can we show our utility in some important disease? Then I think this polymorphisms will really take off. And there's a good chance that nano, when you're doing it, some of the forms might be changing, and people may not even be characterizing it that well with respect to some polymorphic changes in nano. They may not. So they are mostly focused on size and at the most the charge of that one, right? It's a lot of times. So, but if we really want to target certain diseases or show some value in it, I think it'll really take off. Otherwise, if we just increase the solubility or decrease by using some forms, by modifying, or which are, we know that thermodynamically unstable, but now people are stabilizing with excipient and processes. But if it really has to shift the gears between what firms have been doing and what they want to do, they need to find some niche value in certain critical condition. Now, all these, there are a lot of efforts in COVID these days. I'm just thinking, right? So what is polymorphism? What can polymorphism do to formulate it? There are no medications, no vaccines for children yet, right? So can something be done like that, or something to focus on the end user and how polymorphism or areas related to that can be beneficial? The second thing, the important use, these days, they're trying to have online monitoring of things. For example, you want to look at some content, or you want to look at some size, or they're trying to do some fingerprinting with respect to various composition of excipients through real-time monitoring, right? So X-ray diffraction and techniques like that may not be amenable to that. So I don't know in future if there will be a way to do that. Or it might already be done, but I'm not aware of. But the real-time monitoring could be useful. The third thing, the chemometrics, the approach, I think Ratsuri in his lab and some of our lab folks have also done, and where you're trying to look at the effect of a single ingredient in a component mixture and how a group of peaks going up and down rather than one peak going up and down or two peaks going up and down, right? So chemometrics might resolve these drug peaks from other peaks, right? So there seems to be a lot of need for that one. One more, another thing that I saw was, which is very interesting, was the solid dispersions. They were immediate release. A lot of time you're trying to enhance the solubility and bioavailability, but very effective use of this same technology for control release products. So now they're just by modifying the process or modifying the excipient, now they have products that are actually approved as control release. They have very creatively used technology to make control release. So now you have extended release solid dispersion. And there could be other variations there in it component. I think Anne mentioned binary, ternary, and things like that. So you might be having 1 drug, 2 drugs, 3 drugs like that. So these are some opportunities, but eventually how it benefits the patient and what we can do as physical scientists that others are unable to do, that will be the future, bright future. I mean, future is always there, but that will be a bright future for polymorphism.
That's wonderful. Thank you all. We're sort of running out of time, but there's so many good ideas expressed there. I'd like to open up the table again. If you've got quick comments, like 1 minute or so, we can probably take a couple of comments if anyone would like to dive in.
If I could just real briefly, I think USP has to be helped to come along in terms of their standards for physical properties. I think that's a big gap right now.
Yeah. I think one of the main challenges, if you make a product, but I make another product, my polymorph is slightly different, but I'm showing my dissolution is same because I've selected a method that's non-discriminatory or whatever. I'm showing a method that is showing my product is same as yours, and I'm doing bioavailability that's same as yours, then you spend so much on a certain polymorph, but I'm getting my product approved and that's on the market. And a lot of generic companies are going very fast and just bringing their products on the market. If that's not always good and right, that's why we have recalls and other problems later on, but they are getting their products approved.
Yeah, real briefly to answer Karen on that question. In the United States, a generic company can go on the market with a different polymorph if they can show it's bioequivalent. So one of the strategies that innovators have is when they go after polymorphs and other crystalline forms to patent the forms, not just the ones that they're carrying forward, but also defensively ones that they aren't carrying forward, but are still commercially viable.
It's hard to convince small companies of that, though. I have to tell you.
Yeah.
They really don't want to patent all of those forms. And I tell them that generic companies can get very, very creative. They'll go from solid oral dosage forms to patches to very, very different formulations that can take an entirely different type of crystalline form or amorphous form. And my clients are just like, no, we're going to go with oral dosage form. And I'm like, oh, okay. So I totally agree with your point. Trying to get some of these smaller companies, and as you said, some of these patent lawyers who may not know the area, it's hard to convince them sometimes.
And then they have a patent and somebody, they just infringe so easily on that one and citing, this is different. So that's why it's very important to protect that intellectual thing, and which a lot of small firms don't do that very well. And later on, their patents can, people are easily copying that.
Well, that's one thing you can learn the hard way.
Exactly.
And sometimes that's the only way to learn. And sometimes when they do their first due diligence, when they're looking for a partner, and the partner starts asking these questions, well, how is someone going to, because that's a typical due diligence question, how is your solid form patent going to protect against design arounds? And if there's not a good answer for that, then sometimes they come back and go to the lab and find those forms and patent them.
One thing that Mansoor mentioned also, there are some cases where nanoparticles are crystalline or amorphous, and that paclitaxel especially, and the amorphous nanoparticles are claimed anyway to be more active and have better cure, 30% better clinical results, for example, for Abraxane. And that brings one of the areas I didn't mention, but it's really important, I think it's going to emerge. You know, we don't know the structure of the Moderna and Pfizer vaccine. We know there's solid lipid nanoparticles. We know they use 4 different lipids, and they're solid. They're very small. They're suspended, as I understand it, but we don't know the structure. We need to figure out how to get the structure of nanoparticles much more. And this may be where I think it was Ken was mentioning benchtop synchrotrons, or maybe chromium X-ray, short wavelength X-ray tubes to get PDF patterns is certainly going to be one of the ways. But we need to be able to figure this out, I think.
And Steve, that's one of the very nascent areas, really, because the chemistry, I don't think they spent time at all on chemistry. That's why you have a product that you need to store at -80. They haven't formulated.
Exactly.
So
yeah,
it just save it.
It's amazing. We're curing COVID, and we don't know the structure of what we're using.
We don't know the structure. We don't know how to stabilize it. Keep it at -80.
It's objectionable to an X-ray person. Very objectionable.
Well, a year ago, none of this existed.
Yeah,
So maybe,
it didn't
maybe
even
for the boosters,
exist
we'll know
a year
more.
ago.
Well, but not
This
only
is
that, I mean,
objectionable.
they're under emergency use authorization. So Mansoor, when they file their full application, presumably they're going to have to produce some data, aren't they not?
Right. No, but
it
I
can
must
be
say,
pretty empirical. Well, go ahead, Mansoor.
and I was going to say that I must say that considering the time, they have done a great job. There's a great product now.
Yeah, it's really.
If we were waiting for all their complete characterization, we wouldn't have had all those vaccines. So
yeah, well, it looks like they will apply for approval early. It's interesting because I don't know how they're going to complete their clinical trial, and maybe they don't have to. They can use pharmacovigilance data from the general population, but I've read in the newspapers that they're going to be going for approval relatively soon. So I think some of that information is going to have to come out in that approval dossier.
Well, thank you all. We definitely overrun our time schedule here, but it was a really great conversation. So thank you.
Thank you.
So
thank you.
Yeah. Again, I'd like to thank the roundtable members and all the audience for attending this event. And just to mention again, there are links to the members' LinkedIn pages in the show notes. So if you'd like to discuss in more detail the personal opinions that have been expressed today, then please feel free to reach out to the individual members. So I'd like to say have a good day, everyone. And of course, good health. Bye for now.
Thank you all very much.
Thank you, everyone.
Bye now.